Thank you so much, everyone, for joining this meeting today. My name is Jamie Fleward, the head of research at Students Against Superbugs Africa. Today, we are so happy to have Dr. Chris Woodall and Dr. John Mumbo, who will be taking us through our webinar today. And we will have two sessions in this webinar. And the first part of the webinar will be optimization concentration of biocides for mitigation or for prevention of antimicrobial resistance in the environment. And this will be taken over by Dr. Chris Woodall. And without taking much time, I'd like to hand it over to Dr. Chris Woodall to take us through. But before that, I just would like to give a brief introduction about Students Against Superbugs Africa. And this is a youth-led organization that mitigates the threat of antimicrobial resistance within the African continent. And we involve the young people to be the active champions against AMR. And in our organization, we have a very huge representation of undergraduate students and some of the graduate students. And we are so happy to have this educational webinar today. As we learn more about mitigation of antimicrobial resistance through a One Health approach. So without taking much time, let me introduce our first speaker, that is Dr. Chris Woodall. Dr. Christopher Woodall is a consultant at New Microbiology. He is the founder and CEO of Blutest Laboratories, a microbiology contract research organization. He's a lecturer in microbial genetics and virology, Glasgow, Caledonian University, that is Glasgow, United Kingdom. He is a senior research fellow, virology, University of Edinburgh, the United Kingdom. He is a research fellow, neurovirology, University of Glasgow, UK. And Dr. Christopher Woodall holds a PhD in molecular biology, University of Southampton, United Kingdom. Let me hand it over to Dr. Chris Woodall to take us through the first part of the webinar. Thank you and welcome, everyone. Over to you, Dr. Christopher Woodall.
Thank you, Jamie, and I welcome everybody. It's a real honor to be able to support you. I think it's a great cause, and I'm hoping to educate everyone a little bit in that the use of biocides and how they can be effectively used to prevent AMR. So if we look at the actions of biocides, and I'm going to focus on antimicrobials against bacteria, fungi, and viruses. So I'm not an entomologist, so I don't really understand AMR in insects, but I can certainly give you focus on AMR or applications of biocides against antimicrobial agents. So biocides are used on surfaces. They're not used internally to treat infections like antibiotics and small molecule drugs because they're not selective. So they can't be used to specifically kill antimicrobials once they're in patients. So they're used to treat the environment, not a patient. Like antibiotics are used to treat the patient. So they're used on surfaces, particularly inanimate surfaces which are contaminated. These are called fomites. They can be applied in different ways to these surfaces, and the point about applying them is to reduce the antimicrobial, reduce the blood to below levels at which a microbe is pathogenic. One of the most important applications is hand sanitizers, where they're applied as liquids and foams. They're also applied to medical devices by wiping or immersion, or particularly immersion of medical instruments to disinfect them. And the other main application is antiseptics, where they actually are applied onto skin and usually on mucosal or epidermal surfaces. And they're applied as creams, liquids, and wound dressings. So these are the main applications of biocides. Biocides, the main uses are in hygiene and sanitation, and they're used for intervention and transmission of infectious disease. So essentially what they are used for in relation to AMR is to reduce the opportunity for resistant bugs to infect their target, whether it be people or for example, animals. And they intercept the routes of transmission, either the fecal oral route, or the airborne route, or the vector-borne route. So they're important in controlling those particular types of transmission. As antiseptics, they're used to treat infected skin surfaces, wounds, mucosal surfaces, to aid healing and prevent chronic infections occurring in wounds. And we look at the actions of biocides. Different biocides chemically attack microbial structures in different ways. So I've given the examples of how this can happen. Bacterial, if we look at the bacterial cells, but different types of biocides either attack them with protein denaturation, so they destroy the proteins of the cell, which are essential for the function of the cell. A lot of them disrupt the cell wall membrane, so they permeabilize the membrane, and the contents of the cells just spill out. And they also do that to envelope viruses, which are essentially little bags containing nucleic acids, so they're able to disrupt those membranes. Oxidation is the most powerful type of action of these agents, and these are effective against the most resistant types of agents, which are bacterial endospores and naked viruses. Hello? Hi. And there are other mechanisms like non-specific enzyme inactivation and also protein cross-linking, which is important. And this is due to the action of aldehydes. So in this diagram, and I'm sorry, it's quite a busy diagram, but along the right-hand side are the different classes of microbes and their physiological resistance to biocides. So this is related to their actual structure. And so those at the bottom, envelope viruses, are the most susceptible to the action of most biocides. And then there are vegetative bacteria and fungal cells, which have slightly more resistance. Biofilms are an important mechanism for resistance. These are colonies of microbes that join together and have an extracellular matrix, and that extracellular matrix can cause the biofilms to be about 1,000-fold more resistant than individual cells. So biofilms are an important way of microbes being resistant to biocides. Then there are mycobacteria. They have a waxy capsule around them which protects them, and there is some evidence I'll show you later that shows that they can adapt to a certain extent to be more resistant and more susceptible to biocides. Then there are fungal spores, which have a coating which protects them, and they're metabolically have a low metabolic rate, so metabolically inert. Then naked viruses, these are designed to survive in the environment, so they have a very tight structure, which makes them very resistant to the action of biocides. And then the most resistant structures are bacterial endospores, and then prions. So bacterial endospores have a very tough coating that makes them very difficult to break down and destroy with biocides. And prions are very, very resistant, and the reason for that is because they can be damaged and renature. So they can reform themselves after being treated. So along the bottom, I've shown the various different classes of biocides currently being in use, and the green arrows show what sort of spectrum of activity they have against the various classes of microbes. So on the left-hand side of the phenolics, iodine, biguanides, and quartz, these these are effective up to about mycobacteria. They're used as antiseptics, and they're used as a substitute for alcohols for non-alcoholic hand rubs and hand washes. So they're a really important group. Probably most biocides are actually in this group. Then there are zinc, heavy metal ions like copper and zinc. These can be effective against up to including naked viruses. So they're another important group, particularly copper, less so zinc. And treated surfaces with copper and zinc can be effective antimicrobial surfaces. Lactic acid is a good example of the organic acids. These can be effective up to naked viruses. So another important group, and an effective group of biocides which have good activity and no toxicity. Then there are the more established biocides, including ethanol and isopropanol, so the alcohols, which for 70% ethanol is well known to have for its activity against bacteria in particular, and they can be effective also against fungal spores and some naked viruses up to about 80%. So these are fairly effective. They've been around for a long time. Another effective group are the aldehydes. This used to include formaldehyde. Formaldehyde is carcinogenic. It's largely been excluded from use, but it is very effective. You might be familiar with formaldehyde. But also glutaraldehyde. Glutaraldehyde is a pretty good antimicrobial agent. It's a cross-linking agent, and it destroys proteins. And then right at the top here are the sorry, I don't know how to get rid of this. Are the oxidizing agents, and these include peracetic acid, chlorine-releasing agents like hypochlorite, hypochloric acid, peroxide, and other chlorine-releasing agents. So these are the really most powerful oxidizing agents. The disadvantage of them is they're relatively unstable. So although they're very effective, they are also unstable, which means that they're more difficult to transport and use. They've got relatively short lifespans. Okay. So antimicrobial resistance apart, this is how the innate physiological resistance to biocides. Where we see adaptive antimicrobial resistance is most commonly, or at least that's where we've looked, is most commonly in the vegetative bacteria fungi, where there are adaptive mechanisms for antimicrobial resistance. But we'll see. I'll just review that a little bit later about the sort of studies that have been done and what's been found. In terms of balancing the use and application of biocides, they're not selective in action, and they can be very toxic. So this means that we have to be very careful how they affect both the environment and humans in terms of toxicity, or animals as well in terms of that type of toxicity. And this limits the concentrations at which they can be used. And so there is a balance between efficacy, which is the measure of how effective a biocide is, and the environmental impact and ecotoxicity of a biocide, and the human or animal toxicity of biocides. And in this equation, where we look at the relationship between efficacy, human toxicity, and ecotoxicity, we want to enhance efficacy as much as possible and reduce as much as possible ecotoxicity and human toxicity. And that's really why we try and effectively formulate biocides so that they're really good at killing bugs, but they have a reduced effect on the environment. And that's really important because these are not just acute effects, but they're cumulative effects. So over a long period of time, biocide use can have a significant effect. So when we look at biocide efficacy, biocides are best used at the in-use concentration. And in the in-use concentration, this is a concentration that we would normally apply the biocide, and this is a concentration that the biocide should be applied, and it shouldn't be lower than this. The design is essentially to have an overkill. So multiples of laboratory determined minimal lethal concentration are used in the in-use concentration. And the point of this is that they intend to be rapidly lethal to target organisms. So this overkill then is very effective at overcoming any type of resistance that these organisms might have, whether it be physiological or adaptive or selected in AMR. So that's really why biocides are very, very important in treating and being effective against AMR. Is because at the high levels in which they're used, or high level but safe levels in which they're used, they give very little opportunity for antimicrobial resistance to build up. Now, the way that we ensure that this is the case, that the in-use concentrations used, is that we use biocidal efficacy standards. So these are based on laboratory tests. So they're accredited. It doesn't have to be an accredited lab, but normally it's an accredited laboratory study. It's on formulated products, and not just simply on actives, but on the product that's been formulated. So product as it's going to be used, and they're tested at the in-use concentration, and they're tested against representative microbial strains. So these can be, for example, bacteria, viruses, or fungal strains, depending on the type of claim being made for the product. And these tests are carried out under controlled conditions in the lab, and in terms of temperature and soil load, and the reason why soil load is used is because that can inactivate some, but not all types of biocides. So it's important to have a test that's realistic to the actual use of the biocide. And what the standard does is it specifies an X log kill. That can be like 5 or 6 logs for bacteria in X minutes, which can be anything from 30 seconds to 5 minutes for bacteria. And the idea behind this is that it measures a standardized efficacy. So and basically every biocide that has formulation, when it has passed the standard, we know that it's going to be effective as a biocide. So it's about having formulated products, formulated biocides that kill quickly and rapidly, and that's why they should be effective against the opportunity for AMR to develop. You don't need to remember all this, but I'm just giving examples of efficacy standards. Now, these are normally on the directions or on the side of they should be displayed either on the information supporting the biocide or on the label of the biocide. And these are examples of what to look like. I mean, I've actually used examples of EN standards, but the other ones are AOAC or ASTM standards. What these standards on a biocide mean is that it will do what it says it will do, and that's important. So the EN standards are divided into food, domestic, industrial, and institutional types of standards, and these include, and then there are different types of standard for bacteria and fungi, for viruses, and, for example, for spores, but they're more resistant types of organisms. Not all biocides will be able to be capable of all these different activities, most biocides will be effective against bacteria and fungi, but many won't be effective against endospores and viruses. There are similar for medical type of biocides. There are a different set of standards, and these also include effectiveness against mycobacteria, which I mentioned earlier, are a little bit more resistant because they have a waxy capsule around them. And these standards, they have slightly different conditions to represent their use of biocides in a medical environment. And then there are veterinary standards, again, another set, depending on the type of bugs that are being claimed against. So bacteria and fungi, again, mycobacteria and virus. In the case of veterinary, the temperature at which they're performed is, for example, lower than for medical, because veterinary disinfectants can be used outside, biocides can be used outside at lower temperatures, and the soil load is much higher because that represents how they're actually used. So the point I want to make about this is that the way to ensure that the biocide you're using is effective is to ensure that it has the appropriate standards and it's displaying the appropriate standards. Now, what I just want to give you an idea about now is formulation. So, and choosing the right biocide, because that's important for choosing the right biocide for the right purpose. And so the reason why formulation is important is because it can enhance activity, but at the same time potentially reducing the concentration of the biocide itself, so that reduces its toxicity and environmental impact profile. So it's important to try and improve the efficacy by adding co-formulants. And this can include combination of synergistic antimicrobial actives. So this can enhance activity, of course, but formulation, you can't simply just formulate different types of actives and hope to get a better activity, because some actives can act against each other. It's not wise, for example, to mix an oxidizing agent with another type of antimicrobial, because it will probably actually just inactivate that other type of microbial and be used up activating that other type of microbial. Co-formulants. So it's important that formulation is done in a way that creates synergy. And one of the best examples of that is probably mixing different types of quartz together, which enables them to be effective against naked viruses. For example, normally they're only effective against bacteria. Co-formulants can also improve the contact, to improve contact. So surfactants are normally used for surface disinfectants. Foaming agents can be used for hand disinfectants, and they improve the contact by simply allowing the hand disinfectant to stay on the hands, not be washed off, or not to drain off. So they improve contact that way. Sometimes anti-foaming agents can be introduced as well, because foaming sometimes isn't desirable when biocides are used. But in some cases it is, some cases it isn't. So it's about understanding what is the best type of formulation to be used and what type of primary active is the best to be used. So the design of a formulation depends on the intended application of the biocide. Is it, for example, a surface disinfectant or a hand sanitizer? And what it should do is it should improve the effective concentration of the biocide, and so increase its efficacy and reduce its toxicity. If we look at evidence now, to move on to AMR and its relationship to biocides, the studies show there's not a lot of evidence from AMR, and that's largely because biocides are used at overkill concentrations, which and because of the nature of biocides, where they attack multiple sites in the organism, and they're not selective. They really just destroy the fabric of an organism, destroy the structure of an organism. There's not a lot of scope if they used correctly at the right concentration for AMR to develop. But there are mechanisms, known mechanisms for reduced susceptibility in bacteria. There's not a lot of studies on other types of organisms, like, for example, fungi or viruses, but there is potential for them to have for those types of organisms to be selected for their antimicrobial resistance through the use of biocides, but I've not seen any evidence yet for that. Obviously, there are ongoing studies. If you look at the mechanisms by which there can be resistance to biocides, these are multi-drug efflux pumps. So these actively either using ion gradients or ATP energy sources expel a broad range of unrelated and structurally diverse compounds, but these can include major biocide classes. The downside of that is that that also significantly affects the fitness of the bugs that are adapting in this way to the biocide. So it causes the bugs to be like ill. They're not really very. They're crippled. They're not really very. They're much less effective, much less potentially pathogenic, even if they adapt. There is evidence that these can exist and they can expel biocides. And then there's cell membrane permeability. So in mycobacteria, they can adapt their waxy cell wall to make them more resistant to biocides, and they can couple this with a narrowed cell. So that is if you like, an induced physiological resistance that we can see has been seen in mycobacteria. The other resistance mechanism that has been seen is self-surface hydrophobicity of both gram-positive and gram-negative bacteria. So this reduces the ability of hydrophilic biocides to penetrate the cell wall of bacteria. So there are mechanisms that are effective, can be effective against biocides, but if biocides, just to reiterate, if biocides are used at their effective concentration, and then there's significantly less scope for these being effective resistant mechanisms. So the other way in which biocides can influence AMR is by essentially selecting bugs that are resistant to other types of drugs. And this again is in bacteria. So there are two mechanisms: selection or induction of cellular mechanisms that are protective against multiple classes of antimicrobial agents. By applying at biocides at maybe a sublethal concentration, they can induce or select for bugs which have a greater ability to have a general mechanisms or reduced mechanisms of general drug resistance, which can then enhance AMR against small molecule drugs and antibiotics. And then the other way in which that can work is that biocides, again at sublethal concentrations, may select genetic determinants for resistance, and that these are linked to antibiotic resistance. So there is an induced genetic component, potential induced genetic component. When we look at actual studies, I've given two examples. There are ongoing. There's quite a lot of interest in this currently, but in the first study example, I've shown hospital-derived antibiotic-resistant strains of Staphylococci and gram pathogens did not show consistent differences in kill rates, UAC quats, or phenolic disinfectants. So these are relatively only effective against bacteria. But antibiotic-resistant strains, so strains that are effective against have effective resistance against antibiotics, don't apparently have any different resistance against biocides. In another study example, however, hospital strains of Staph aureus and gram-negative bacteria, which are antibiotic resistance, did show minimum inhibitory concentration values for chlorhexidine, cetrimide, and chloroxylenol, which is a phenolic disinfectant, that were above their respective use concentrations. So there is an example where, in this particular study, there is an example that antibiotic resistance mechanisms can also have a protective effect against biocides. It's really I'm come to the end. Sorry, it's a relatively short talk, 30 minutes, so I can't cover all of the various factors, but obviously the way in which you choose biocides and how you use them is important in their effect, them being effective against the opportunity for AMR. So one of the considerations is expense, because biocides can be expensive, but they don't have to be effective. So, for example, hypochlorite bleach is a very effective high-level disinfectant. It's effective against bacterial endospores, and it's relatively inexpensive. Similarly, alcohols can be used, but there are obviously cultural reasons why alcohols can't be used, and so but they're both relatively cheap, effective biocides to use. Then you have to consider what type of biocide, what's going to be used for. Do you need a high-level disinfectant? So ones that are effective against bacterial endospores, or can a less harsh biocide be used? That, for example, doesn't cause skin irritation, but is still effective against bacteria and can be supportive in that way. So skin irritation is an important factor, obviously, in antiseptics and in hand disinfection products, because that reduces what's available. And these are generally the lower level biocides, like quats at low levels, chlorhexidine, for example, triclosan can be used for that purpose. So it's important that then that they're used for that reason, and surface disinfectants can be used, but you've got to bear in mind that these might also be corrosive. So if an agent is corrosive, it can't be used, and that is one of the drawbacks of actually hypochlorite, and that's why there are substitutes for hypochlorite, because it can be corrosive. And that corrosivity can give an opportunity for an environment where bugs can survive in microscopic cracks, for example, in surfaces. So that needs to be taken into consideration. The shelf life stability of a biocide is also important. If biocides are used beyond their shelf life, which generally is 1 to 2 years, but for some oxidizing products, it's considerably shorter than that. There's a danger that the low levels of biocide used can induce AMR. And another type of biocide that is commonly sold is one with residual action. So these are placed, put on the surface, and they have a prolonged action over a period of hours. They can be useful, but obviously there is also a potential with these agents that they can potentially induce AMR. Another important consideration for biocides is application methodology. So biocides should be used as part of a cleaning strategy, so that they're used consistently in the most effective way. And there should also be an assurance of good compliance, so that once you have a strategy in place for cleaning and disinfecting an environment, it's important that that is maintained and monitored to ensure that biocides are used effectively, and that they're being used at their in-use concentration to reduce environmental opportunistic pathogens. Another way in which biocides can be used to reduce the opportunity for AMR is to rotate biocides with different actions. So this is commonly done in clean rooms, for example, and it can be done in some hospital environments too. And the reason for this is so that essentially, if there is the potential for emerging resistance, one type active biocide, you can change that biocide to another one with a different type of activity, and it's unlikely that the bug with resistance to the first biocide will have resistance to the second, so it prevents the buildup of AMR. Okay, so that's really it's been a very short talk. I've tried to give you some indicators of why biocides can be effective and safely used at their in-use concentration. They are effective. They're used to treat the environment and reduce the opportunity for pathogens to infect patients or animals or their target. And so used safely and in the correct way, biocides are very effective way of combating AMR. Okay, so thank you for your time. If there is time for some questions, I'm happy to answer them.
Thank you so much, Christopher Woodall. That was a very insightful presentation. Thank you so much. And now we're moving to the second part of the webinar, and we'd like to have our second speaker, that is Dr. John Mumbo. And Dr. John Mumbo works with the National Environment Management Authority of Kenya as the Principal Compliance and Enforcement Officer, currently heading the Chemicals Division with over 15 years experience in the awareness and education, guidelines and regulations development, research and policy in chemicals and environment management. He is the national focal point of environmental dimensions, antimicrobial resistance under the One Health approach. He follows atmospheric and air quality and pollution management, environmental phase of chemicals, hazardous toxic chemicals, and waste management. His affiliations include the Royal Chemical Society, Kenya Chemical Society, Kenyan DAD Scholars Associations, Environmental Institute of Kenya, and the Persistent Organic Pollutant Review Committee of the Stockholm Convention. He is advocating for cancer prevention through awareness, education, policy, and regulatory interventions in order to minimize or eliminate direct and indirect exposure to chemical risk and contaminants in products and the environment that are suspected to be sources of cancer. Dr. Mumbo, if you can hear me, you can take over the second part of the webinar. Thank you, and over to you, Dr. Mumbo.
Okay, thank you, Jimmy. I hope you can hear me well, and I also hope you can see the presentation well.
Yeah, sure. We can hear you. And
Yeah, yeah, definitely. Yeah.
Okay. So I'll proceed, and thank you for that elaborate introduction. So I don't need to say much about myself. You gave me this topic about the monitoring of biocides in the environment, prevention of AMR. I'll proceed to how the presentation has been organized. We look at the introduction, of course, the basic concepts and definitions. What is biomonitoring? We'll also get into biocidal products. I think this has been actually captured well in the first presentation, so I'll not get into much of the discussion. Of course, we'll get into production, use, and fate of these biocides. We want to know how they are produced, how we use them, and of course, what happens when they get into the environment. We've talked about resistance in the previous presentation, so I may not get into that. And then we'll look at what are the actions that we need to do in general in response to AMR. And of course, I'll go specifically to what we need to do about resistance that comes with biocides. And indeed, I'll also just bring some highlights about the need to identify other pollutants within the environment, what kind of environmental action is needed. The presentation is quite long. I've deliberately left some other sections which you'll read later, or maybe time allowing, I may be able to also capture. So they have not been covered in this summary. So I'll not get into, I don't want to assume that we understand biomonitoring, but in this case, we are looking at a systematic measurement of compounds. We can also look at the detection of cells molecules, which have been alterations in living organisms, with the purpose of identifying, we're doing monitoring to identifying or assessing what is the potential hazard exposure or effects with the relation to chemicals. So, it's something that is done systematically, and of course, we are looking at the effect that is causing on the alterations in the living organisms, and what are we looking at? Also identifying or assessing the potential hazard exposure and effects to these chemicals. Also, just on these topics, of course, when you're looking at biomonitoring, of course, it uses bioindicators. You also have to use biomarkers. I've attempted to just summarize the bioindicators as organisms that can be used mostly for identification. Here we're looking at, what can be used to identify and, of course, to provide a qualitative determination on human-generated environmental factors. We know about the bees. We know about the dragonflies, and of course now we also need to look at the biomonitors. These organisms that can be used for quantitative. The first line is qualitative, but here we're now looking at the quantitative determinations of these contaminants. This can actually get into the levels. So we want to look at how is the accumulation happening. We want to look at to study these organisms in detail. So there are those ones which are indicative in nature, and those ones that can be used for monitoring purposes. And then I'll proceed now to also look at the techniques. I'll mention that I think we all know about bioaccumulation. There's also the biochemical alterations. There's morphological and behavioral observation. We also need to look at the population and the community level approaches and modeling. So just to go into brief description of these techniques, of course, bioaccumulation, this occurs when the absorption of the toxic substance is greater than the way it can be eliminated from the system. And after this accumulation happens, of course, this results in biochemical alterations. In this case, we are looking at the interactions between the pollutants and the biological macromolecules within your system. These are the proteins and the enzymes which can start causing changes in your neurotransmitters, immune systems. And then this, we can also look at it also from another point, whereby these are actually direct morphological and behavioral, whereby you're looking at the structural damage, you're looking at how it affects the behavior subsequently, and this actually, the action of on the application, and then. We also need to look at the population and the community level. Here we're looking at when I mean the population, I refer to the density and size distribution of these organisms within the environment. We're also looking at the species richness. We are looking at the variability, the composition. You can look, for example, at the chaotic system, which is being affected by metal pollution. We're talking about heavy metals. How does this kind of pollution affect the richness and the population of these kind of organisms within that environment? So in addition, apart from these other techniques, we can also proceed to do model experimental results or published data. These results actually help us to develop mechanistic models, and based on the variables that have been collected from this experiment, it can be used in different situations, assuming that the same variables and factors are applied. So it's really important when you look at these techniques, when you're getting into modeling in the context, like for example, here in Kenya, do we have data? Do we have results that can actually help us to use this in monitoring? We also proceed now, after covering the basic concept. I'll just I think this one was actually covered better, but just for the concept, since I'm speaking from a regulatory point of view, I would want to look at this just to look at how we define this when it comes to regulation apart from research. We look at these biocides as, these are active substances. They can also be preparations containing one or more active substances with the intent to destroy, to render harmless, to prevent the action of, or exact a controlling effect. And the ultimate goal is the target is on what we perceive as harmful organisms, and this can be done through chemical or biological means. So it's really important when you're doing biomonitoring of these biocides, you need to understand why are we looking and why the biocides. In addition, now from understanding what these biocides are, we need to look at also now, when you look at the production, production relates to, we are looking at the quantities. We want to look at the use, and we also want to look at how, after using, what happens to them in the environment. But you can see from my presentation, biocide is actually a class of chemicals which is actually not regularly monitored. I think we all know here in Kenya, it's even categorization of these biocides is actually quite a big problem, because the Pest Control Product Board deal with pesticides, and the pesticides is a class on its own. Biocides is also a class on its own, which, as we speak, does not have maybe guidelines or a regulation. And since it is not monitored, the amounts of the products applied or used, it's actually quite unknown in the context of Kenya. Though as NEMA, we issue permits for some imports, which you can now say, there's some companies that are actually import them for their use. So the production volumes are considered significantly higher than those of antibiotics. From this, we can actually assume that the huge amount of biocides in the environment may actually, of course, induce a biological hazard. Of course, when applied to the material, this is just basically based on the on assumption, because there's no data to support that. Now we'll now go into the uses. I'll just talk in their broad uses, just in not into detail as was done earlier. Here we find that is used in the healthcare, of course, as for the contamination process of healthcare, environmental surfaces. Some examples have been given here, which were mentioned earlier. They are also used in the consumer products. And here, when you're talking about consumer products, we refer to building materials. We look at consumer products such as cosmetics. We look at cleaning products, disinfectants, wipes. They can also be used in furniture, curtains, and so we normally use this a lot. We export a lot of this as Kenya, but this lecture is actually helping us to have a better understanding of what comes with these materials. In addition to that, they can be used in food production. Again, also here is for disinfection of the production plants. Production plants here, we mean the plants that are actually used for preparing these foods, like fish processing plants, food processing plants, and their containers, basically to control microbial growth in the drinks, and also the contamination of carcasses. Here we have the candidates like chlorine, ozone. They can also be used in animal husbandry. We all know that when, of course, to protect these animals from endemic and zoonotic diseases, we know when we go to farms, there's the cleaning, disinfection. We have foot dips. We also have vehicles that also get into being disinfected during outbreaks. Of course, direct application on animal surfaces such as teats. Before milking, we look at preservation of surfaces, and also of eggs, even semen, and there are quite a number of examples which are actually used. And again, we also need to understand that they also have application in environment, and in this case, they have application in water treatment. We know about chlorine. They can also have a treatment in water treatment. There's industrial use. I know in Africa or in Kenya, we don't use a lot of cooling towers, but where there is a lot of hot water generated, like in winter, these are actually used for disinfecting, against what is called Legionella species or Legionellis. This actually thrive in that environment or those taps. In addition to that, they can also be used as anti-fouling agents in building, on antimicrobial surfaces. They are also used within surfaces and to protect the surface. So as we can see, biocides actually have quite a wide range of application. This means that now their dissemination into environment can also be quite significant, and this leads me now to resistance. I'll not get very much into this discussion because it was discussed earlier, but of course, occurrence have actually been detected, which have actually been reported as early as 1950s. Here we are looking at, in some instances, these resistance were emerged because of improper use and also or storage, as was highlighted earlier. The issues about when you use the application method, the issues about sublethal dose, so this the resistance is now becoming clear about how we use it and how we store. And then there's also in some cases whereby, like in the hospitals, they're also effective against nosocomial pathogens growing as biofilms, which was actually mentioned earlier, and attached to surfaces and fail to control. Of course, there's also this tolerance to salmonella by this antimicrobial in the biofuel, which was attributed to low diffusion. Yeah, so as was, it's basically coming that we really need to look at how we can apply this biocide so that it can be able to go beyond these barriers. And I don't want to talk very much about mechanisms, but what was alluded earlier is that biocides have multiple target sites. It has a multiple approach of how to deal with these microbial cells. And what's coming out clearly is that the resistance is due to decrease of the intracellular concentration of the biocide. As long as the target does not get enough concentration of this biocide, of course, they build that resistance. And this principle is supported by, we are looking at the changes in the cell envelope, which was described earlier, waxy, change in permeability, efflux, how the organism excretes or removes this or degrades the same. We proceed now to look at still, when you look at this resistance, they are actually intrinsic or extrinsic internal and external factors. Like, for example, these intrinsic factors, this is based on this biocidal agent. We're looking at the agent itself and its application. You see, there's the contact time and also the concentration. Of course, if it is less, you expect the contact time, there's this about the issues of the residual effect. We're looking at also at extrinsic factors, which derive from environmental bring up from environment. The temperature of the environment is important, as most substances have a lower efficacy at lower temperature. And also the presence of proteins also reduces the efficacy. This was actually described. We talked about efficacy in the previous presentation. And in addition to that, we also look at the mode of contact also influences the efficacy, as does the contact time. pH is an important factor. The concentration of the microorganisms, the age of bacterial community, and the protection by attachment to particulate matter. There's also the presence of the biofilm, which was actually mentioned earlier, is playing an increasingly role in building this resistance. So we now have a knowledge about their uses. We have seen how the resistance come about and the factors that attribute to this resistance. Now we really need to look at what is the action that we need to bring. We need to strengthen environmental action because it's coming out clearly that AMR development, transmission, and spread in the environment can be attributed to microbes. Like, for example, in bacteria, here I'm just talking in general. Yeah, you are looking at bacteria in water. It can be in soil. It can be in air, which can develop resistance following contact with other resistant microbes. Of course, as from the previous discussion, we see that this bacteria can build resistance. And once build resistance, now when they're communicating amongst themselves, they start now acquiring these genes, resistant genes that can also start causing them to be resistant. We've just described, our topic today is about pollution by chemicals with antimicrobial activities. There's the pharmaceuticals, certain metals, and also the biocides and other compounds. In addition, you also have pollution containing antimicrobial agents. Yes, this pollution can come from wastewater streams, as highlighted earlier, hospitals. We also look at agricultural, with the pesticides, antimicrobials, and chemical manufacturing. There's a whole range of what gets into the environment that actually causes AMR development and also the resistance. So this, we need to look at it. It's like, as we are looking at biocides, we'll not only find biocides alone, we'll also look at other factors that actually also lead to this resistance. So this actually leads us to the need to identify pollutants that exacerbate AMR in the environment and their sources. I bring this because I know we are talking about biocides, but we really need to understand why are we using biocides, because there are sources that actually cause these, that actually bring these microorganisms, such as poor sanitation. We look at sewage, there's wastewater, there's also the issues of effluent and waste from pharmaceutical company, effluents and waste from health care facilities, uses of antimicrobial, of course, in crop production, and then releases and effluents in amino production. Because this here, the purpose of this slide is actually to show you the application of these biocides. It's really quite big and can actually help us to appreciate the scope that can actually also lead us to how can we start doing undertaking monitoring of the same. Now going specifically to biocides, I know we were looking forward to looking at methods on how to do bio monitoring of these biocides, but before you do monitoring, we need to understand that the data on the fate and the concentrations of biocides in the environment is quite sparse. So there's really need for a baseline that can actually help us to appreciate how these biocides end up in the environment, their fate, and also the concentrations. In addition, there are also no validated methodologies are available for the determination of dose-response relationship and of the threshold triggering the emergence of antibiotic resistance or a selection of resistance. So that's why I was looking at it as opportunities, the opportunities to help us get this information and get the methodology. And in addition to that, there's also lack of information on production and use volumes, which limits studies or assessment on exposure of bacteria to biocides or their metabolites in various matrices. And again, there is the need for environmental studies focusing on identification and characterization of resistance and cross resistance to antibiotics following the use and misuse. So, with this kind of very essential information lacking, it's like you really want to get a baseline, you really want to get the pattern, you really want to get certain behaviors that can be able to help you now to come up with the correct tool to be able to undertake the monitoring. Just in previous slide, if you don't have data on the fate, you don't have the data on the concentrations, how will you be able now to start doing monitoring of the same? And that's why it's also quite good that we had this lecture, so that we're able to understand their production, their uses, which can help us now to start focusing our area on actually to do collecting data and also to determine concentrations. And once we have this, to burn out, we got to monitor the trends of the same within the environment. But away from that, of course, the environmental action is actually quite needed. We need to enhance environmental governance. Here, I mean, we need to have laws and regulations on this biocide. As I've said, in Kenya, we don't have it. And we also need to do planning. And planning here, we are looking at environmental planning in the sense that in the in the sense when we are undertaking a national environmental action plan, when you're looking at the hotspots, we also need to plan on how we can deal with this slowly emerging problem. And there's also the need for regulatory frameworks. We also need to identify and target priority AMR relevant pollutants. Of course, biocides is now becoming one. And then we need to improve reporting, and which will lead to surveillance and monitoring. We need to educate our politicians. We need to educate our leaders, professionals, even through the social media, to be able to appreciate that this subject is really important, and it's really bigger than what we really thought it was. And you know, this should actually lead to budgeting, financing, planning, and of course innovation and research, and developing methodologies, standards, and training more people, you know, to be able to be experts in this field when it comes to biocide. This is actually what I'd mentioned earlier. I'd intended to stop my presentation here because the others you can follow later, which talks about environmental dimensions of AMR governance in Kenya, where NEMA is the institution that deals with the governance of all policies that relate to environmental management. In addition to that, I've picked a few of AMR applicable environmental regulations, which, for example, we have environmental impact assessments and environmental audit. This is the water quality regulations. We also have regulations that deal with waste management. We also have regulations that deal with air. I know somebody will be wondering, how does air mean? This, of course, most of them are aerosols. The pharmaceutical industries, of which actually also produce emissions. So we cannot ignore air. My presentation has actually gone to open up like the environmental impact assessment, whereby, for example, there's the aerial spraying. We also have different areas of agriculture that actually rely on fertilizers, pesticides, and then it proceeds to talk about sections of schedules of the projects which require mandatory EIA. These are like the waste, especially for the waste disposal. We talked about effluent, we've talked about wastewater treatment. We need to talk about dump sites, because dump sites is actually where you'll actually end up dumping unused biocides. And then what do we need for environmental impact assessment? Of course, we need to develop guidelines to aid in the review of environmental impact assessment reports and environmental audits, such like when somebody is developing an environmental impact assessment report for a poultry farm, you should not forget to include the monitoring of these pesticides. When you are developing an EIA of one hospital, you need to look at how the range of the chemicals are and antibiotics, including biocides, and how they're supposed to be managed. We also need to train our experts on the subject of antimicrobial resistance to be able to report this effectively in these documents. There's the water quality regulations about the issue standards for discharge into the environment. Unfortunately, we don't have one covering biocides. The other parameters that deal with the microorganisms, and there are quite a number of actions that needs to be done on this water quality, because we need to map key sources of contaminated or high-risk facilities that have an impact on AMR. Of course, we also need to do enforcement. There's also the waste management regulation. What are the provisions? These pharmaceuticals are classified as hazardous and medical waste. Incineration is the most preferred method. And then, of course, there are quite a number of actions that also needs to be done here. Of course, we need to develop and disseminate guidelines on procedures and protocols of waste disposal for different sectors. I think the presentation now ends up there. Here we are looking at a One Health approach, whereby it's not only the health and the animal health, but you also need to have the environmental health in dealing with this in the management of biocides. Otherwise, thank you for listening. Back to you, Jimmy.
Thank you so much, Dr. John Mumbo. That was very informative. very educative indeed, and I really appreciate that. And we have learned a lot, in the nexus between environment, biocides, and antimicrobial resistance. And that was very important in this discussion. Thank you so much for that discussion. Let us move now to our next part of the webinar, that is the Q&A session, and they can see 2 questions here. If we have more questions, we can let them come in as we wind up the session. And one question from Mariam, she's asking about, and I think this one goes to Dr. Christopher. Are there specific regulations to prevent anti-biocidal resistance, or the selective pressure they may cause due to sublethal concentrations? And the second part of the same question reads, are there feedbacks mechanisms that informs the current efficacy of the biocides and how to manage any ineffectiveness of the biocides? I think so. So, Dr. Christopher, I hope you got the question. Over to you.
Yeah. No, well, the simple answer is no. The way in which we currently use biocides is to ensure that they're effective at the point of use. But there's not a lot of legislation looking at the fate of biocides and the biological impact. Although it's not particularly my area of expertise, but there are requirements for environmental impact assessments to be done, but apart from that, there's very little legislation in relation to biocides and AMR. I think so. It's really the answer is that there's not very much, certainly in the UK and Europe, relating to the impact of biocides on AMR.
Okay, thank you so much, Dr. Chris. We have another question still from Mariam. She's asking about, and I think this one also goes to Dr. John Mumbo. From your experience, do biocide use in industries such as oil and gas, that is pipelines, have a bearing in fostering selective pressure and subsequently AMR in the water ecosystem? And are there any monitoring guidelines for this, that is both oil and gas industry and the biocide manufacturing industries? Over to you, John.
Okay, just as I mentioned earlier, and I think we also need to know that the subject of antimicrobial resistance is actually quite new and also emerging in the context of Kenya and also the developing world. Yes, much of the biocides that we issue permits for are usually for disinfection, and they're usually used for control purposes, for medical purposes, but they don't have they don't have the intention, or they don't have the knowledge that this can actually lead to some form of resistance, or the microorganisms that they're using can actually cause them to look not effective. Normally, what happens from the experience, I think you all know, when you know that one of these biocides is not effective, you'll go for another range that looks that will actually deal with it, and will actually try to overuse and even misuse. Yeah, but we've not actually gone to the extent of actually trying to find the consequences of actually overuse or misuse, or trying different ranges for the same problem. It's the subject that actually caused me to think seriously, because we are actually at the moment reviewing our regulations, and one of the regulations that deal with this is water quality regulations. What we've done is to include in the monitoring aspects, the monitoring of antimicrobials. We didn't want to just mention antibiotics, but antimicrobials, so that the intention here is to come up with guidelines that can guide and that can educate first and provide guidelines on the usage of the same, and also for monitoring the same. We also have our chemicals regulations. It has been quite a challenge to see how to introduce biocides in that regulation, because it deals with industrial chemicals, and the agency that deals with pesticides has also not done much to, or not done much, as does not have provision on how to deal with biocides. So it's also a space that we're also looking at. Since we issue permits on the same, we're also looking at how we can develop guidelines for the same, and we are looking very carefully at what the EU is doing, because they have a standalone regulation that deals with the biocides, and you know, for that class of chemicals, and we want to see and also borrow from the experience, especially on the concentrations or the standards that are eventually allowed into the environment. Yeah, so that's actually quite a gray area on, especially when it comes to standards, because for a regulatory agency, it's really important to tell when a chemical is being misused, or where pollution is occurring, or where harm is being caused. So it's something that we can also look at together. Thank you.
Yeah, thank you so much, Dr. John Mumbo, for that elaborate answer. And I think also, yeah, something that you also mentioned about scarcity of data in terms of biocides research, monitoring and regulation of the environment. I think, yeah, maybe even, I think there's a lot that still need to be done in terms of biocides use and regulation, because even apart from AMR, there are a lot of other health issues associated with the use of biocides, especially in the environment, which are directly linked to human beings, animals, and we can say planetary health. I think there's still a lot to be done, especially in Africa, the continent, whereby I think there's a scanty of data compared to other parts of the world. So I can see Vanessa, your hand is up. Vanessa, can you hear me?
Yes, Jimmy, I can hear you. Can you hear me?
Yeah, sure. Can hear you. Go on, please.
Okay. So thank you, Dr. Christopher and Dr. John, for that presentation. So I have a few questions. My first question is to Dr. Christopher. When we talk of biocides in the AMR perspective, I think we've come to a point where we are actually seeing a lot of pathogens out here they have genes that are not, like Dr. John was saying, that are not intrinsic. So most of them have virulence genes that encode for resistance against this biocides. So now my question is, considering that for most of these particular pathogens, the resistance is extra chromosomal, would there be a prescription where we would say that because this particular bacteria has gained resistance to, for example, ethanol at this percentage, would we have a recommendation where we try and increase the percentage, or how do we actually curb that? How do we curb the fact that most of these pathogens have plasmid encoded virulence genes that are now transferable from one organism to another, and we cannot literally say that we would do away with the biocide? So how would we handle this resistance?
Yeah, I think there's two ways. I think the first way is to use biocides at their appropriate concentration. So that is an overkill, and you'd expect that to be effective. So it's important how you apply biocides, not just what you use, but how you apply them. So it's important to apply them in the right way. And in addition, what you can do is you can overcome that resistance by rotating biocides, so using different different types of biocides, because it's likely that the same resistance would be able to be effective against the different types of activities of different types of biocides. So that's the strategy you would use in that type of environment to overcome the resistance that you might be experiencing.
So now, for example, in hospitals, now we have as Dr. John was saying, nosocomial pathogens that are mostly multi-drug resistant. How would we try and curb the fact that we cannot even use antiseptics anymore, or even if we use them, they're not as effective as they used to be because of the virulence genes? Would we just spray everything? And also, I'm thinking in terms of fungi, I really don't think it's easy to get rid of fungi with just antiseptics. So what would, in your own recommendations, how would we, in that particular setting, how would we handle that?
Okay, fungi are slightly different in that they produce spores, and spores are quite resistant to most antiseptics, because antiseptics are normally lower spectrum biocides, and they're more effective against bacteria, for example, than they would be against fungal, for example, coniidiospores. So the only resort you have when using biocides, if antiseptics are not effective, is to try and reduce levels in the surrounding environment. And you can do that by using harsher types of disinfectant to treat surfaces. You can use zonal disinfection using aerosolized disinfectants. That's the only recourse you have if the antiseptics you're using are no longer effective.
Okay, great. Thank you so much for that.
Sorry, it can't be more helpful than that. But
no, that's okay.
I think that's the only course of action that can reasonably be taken.
So if I'm getting you right, what you're saying is if we're dealing with antiseptic resistance, the only thing we can do is basically up our percentage or reduce what we're using to limit resistance, but we cannot actually come up with a new, like when you think of antimicrobial resistance, we cannot actually come up with new therapies. It's either we up concentration or reduce it.
Or reduce the opportunity for infection in the first place by treating the environment with biocides. I mean, there will be emerging technologies, but for new antiseptics, but these take time to develop, and we don't have a pipeline of new antiseptic actives available, and so at least in the short term, it's necessary to resort to actually better hygiene and disinfection to reduce environmental opportunistic pathogens.
Okay, thank you so much.
Okay.
I have another question for Dr. John. So my question to you, Dr. John, is I've heard you talk about the multiple regulations we have in Kenya for handling the spread of the use of these biocides in the environment. But I've not actually heard you particularly talk about its use in the farming sector or just agriculture per se. And when we think of AMR, again, the whole One Health approach is when the health of one part is at risk, then the health of all might be at risk. So when you think of it in that regard, when you look at it in the fact that there's a lot of biocides, just leave alone antimicrobials, biocides that are used in prevention in agriculture, is there like a way as NEMA you are trying to regulate that, or I mean we just trust farmers and we hope for the best?
Okay, thank you very much. It's quite a very interesting question in the context that what I can say, the agricultural sector has really been ignored. We've always been looking at that sector as just producing food and also sustaining the economy of this nation, but we've not actually looked at what goes into bringing causing the food production. What you're doing quite a lot, you're working a lot with the Ministry of Agriculture. They've actually come to the realization that they really need to do a lot, especially when it comes to the management of the chemicals, the management of antibiotics. There's some which are actually expired, there's some which are actually need to be disposed. We're also looking at, let me give an example of when they do application against there's vaccination, there's also the treatment of cattle. There's this practice in Kenya whereby when a cattle, that a dairy cow is treated, you have to allow the milk to drain before you can take it to market. That is not actually being observed. There's also the issue of also using hormones in fish production, and some of this becomes really very sensitive. For example, the case of hormones and fish production, the sector doesn't actually want to hear anything. Okay, they don't want to hear controls because they want to stimulate food production, but at the same time they also want to use hormones which can actually also find themselves indirectly to us. So we are engaging and creating awareness to the agricultural sector. I think you also understand about the issue of effluent that come from slaughterhouses. We are also looking at when you've already applied pesticides or biocides, and then you use the same manure for application in the farm. Yeah, all these quite a number of strategies that we're actually going to use, and the regulations are actually coming to place. They are basically, we don't have non-point sources, we have 4-point sources. Point sources, I mean like effluent that are being discharged into the environment, that is for like from discharge points from large poultry farms. There's the need for environmental impact assessment, and that's why we were very keen that we want to have our environmental experts and environmental so that when they undertake environmental audits, they don't just assume that everything is all fine within the agricultural sector. There's also the issue of managing the waste that come from this sector. We developed a guideline on how to deal with veterinary care waste from the veterinary sector. It has been finalized, but it's really, you can imagine just the veterinary sector. For us, it was just dealing with only chemicals from the veterinary care of these animals. So it's really something that we're trying to see where to start from. But what I can assure you is that we've reviewed our regulations. Like there's the waste management regulations, there's the chemicals regulations, we also have the water quality regulations, and we're also going ahead to come up with the guidelines. On Monday, we shall be launching the National Action Plan on Antimicrobial Resistance, and one of the guidelines that we're actually targeting is to come up with the guidelines for managing this kind of waste. And when I'm talking about waste, I'm not talking about waste, because waste fall in different categories. There's the organic, there's hazardous. It covers all of quite a number of it. We also need to do inspection. As you can hear, there's really a lot that needs to be done for the sector. It's just a matter of sitting down and looking on how to prioritize what needs to go first as we see how to handle this resistance within the agricultural sector.
So, okay, that means there's hope. But the other question
of course, yeah.
You haven't really mentioned anything about antimicrobial stewardship. You've talked about you want to deal with what comes after, when these people use that and farmers utilize this and waste is that in play. But what happens? Have you, as the board, have you actually thought about educating and making sure that farmers are aware that you have to be careful with how you use antimicrobials?
Yeah, we have a strategy, and we've actually divided our roles. When it comes to stewardship, that one we've left to the main actors within the sector, that is the agricultural and that's for animal health, and also for the for the health, that is the human health. Yeah, because they deal directly with this with the clients and also with the animals. And I can also wish to inform you, maybe in the course of next week, after we've launched the National Action Plan, I can share with you the document so that you can be able to see how this how the aspect of stewardship is actually being handled within this strategy. Yeah.
Okay, thank you so much, Dr. John. Again, thank you for the wonderful presentation.
You're welcome. Thank you.
Thank you so much, Dr. John and Christoph. It has been really an engaging discussion and very insightful. Thank you, Vanessa, and I hope that you have gained an understanding from that. I think there have been a lot of interesting questions, and I really believe that we can make use of this. And I think another thing that is really coming out from this aspect of biocides is that we understand that biocides are not microbe specific or pathogen specific, and that's why it's also difficult somehow to optimize the concentration of these chemicals for mitigation or for prevention of AMR in the environment. And despite the fact that we're using this in day out activities in hospitals, in the industries, in our homes. So I think that's another very big challenge, and that maybe hopefully moving forward, if a lot of emphasis will be put in this in terms of research, regulation, maybe we hope that this would be improved in future. So I don't think we have any other question from the audience, and we just want to sign off our session today. So I just would like to ask Christopher and John to give us final remarks for the meeting. So we can go, we can start out with Dr. John Mumbo.
Okay, thank you, Jimmy, for such an exciting topic. I didn't expect that biocides would actually be that that significant. What you're looking for is to come up with guidelines on the same. Just still wrestling on how to have it included within the regulation, because we don't want to have the environmental sector look as if it's overstepping its mandate by carrying out the mandate of the other institution. But what I can encourage you, since you are scientists in this area, as you keep on researching, as you keep on developing methodologies, what you need to know that the methodology that you use for your research end up becoming our guidelines. So I always remind the researchers and academia that when you are doing your surveys, when you are doing your field work, when you are doing your research, the methodology that you use to arrive at a given results always help the regulators to come up with a guideline on how to monitor the same in the environment. So those ones who are in the space of these biocides, please have a look at it. I'll also try to see how we can start picking up a guideline on the same so that we can also bring it back for discussion. Thank you.
Thank you, Dr. John Mumbo. Over to you, Christopher.
Okay, I would just say it's important just to use biocides effectively and increase their efficacy and try and reduce their environmental impact. And then that's the best way in which they can be used and remain effective. There will be a pipeline of new biocides, and I'm really sorry that the antiseptics currently being used are no longer effective. It's sad to hear that, but hopefully there's a new biocides or new antiseptics will be developed, new types of biocide will be developed, which will help them remain effective in healthcare, certainly. So, and thank you for the opportunity, Jimmy, for being able to speak.
Thank you so much, Dr. Chris. I think, now that we have been pushing this webinar every time, but I'm really happy and glad that you really made this really great time to this webinar today, and I really appreciate that. And this is not the end of webinar series, educational webinars. So we might be reaching out to you anytime soon, as we also consult on you in matters environmental research, biocide research, and all that. And we really look forward to have you in our future webinars. And I really wish to thank everyone who made this webinar today. And we hope to see you in our other webinars soon. And I think up to this far, we just like to sign off our meeting today. And yeah, we meet next time. Thank you so much, everyone, and bye-bye, and keep safe. Bye-bye.